Genetics. This is chapter 11, and we're going to be working with Punnett squares in this particular chapter. First, we need to do a little bit of history on genetics. Gregor Mendel, who lived from 1822 to 1884, was responsible for the laws governing the inheritance of traits. So we'll think of Gregor Mendel as being the grandfather of genetics. Gregor Mendel was an Austrian monk, and he studied the inheritance of traits in pea plants. He also developed the laws of inheritance. Mendel's work was not recognized until the turn of the 20th century. So when it comes to the study of genetics, when a timeline occurs, it's fairly recent that we learn all about the things that are inside of our cells. Between 1856 and 1863, Mendel cultivated, and cultivated means to grow, and tested some 28,000 pea plants. He found that the plant's offspring retained traits of the parents, and once again, he was called the father of genetics. He saw that when he worked with pea plants, that the parents passed specific traits to the offspring. The picture here shows the site of Gregor Mendel's experimental garden in the Czech Republic. And what you are looking at in that picture are pea plants. Okay, the terminology. As you know, in science, the terminology is sometimes confusing, and that is what makes the topic so difficult. Well, it's going to get a little more challenging in genetics because there are so many opposites. So this particular podcast is basically going to go through the different definitions of all of the vocabulary words we're going to use. First vocabulary word is trait, and a trait is any characteristic that can be passed from parent to offspring, whether it's eye color or hair color. Those are traits. Heredity is the passing of traits from parent to offspring, and genetics is the study of heredity. So these are key terms which you need to remember. Trait, which are the characteristics that are passed. Heredity, which is the passing of the traits. And then genetics, which is the study of heredity. Once again, three very important vocabulary words. When we talk about genetics, we'll talk about genes. And these are not the jeans that you wear as pants. Alleles are two forms of a gene. They can either be dominant or recessive. A dominant trait is the stronger of the two genes, expressed in what is called a hybrid, and it's represented by a capital letter. And this term, hybrid, which you're going to see later, is a mixture of one dominant and one recessive trait. The dominant trait is once again represented by a capital letter. Recessive is the gene that shows up less often in a cross and is represented by a lowercase letter. So these two particular vocab words are opposite, dominant and recessive. And each of these is an allele, which is two separate forms of a gene. Genotype. there are two different genotypes. And when we talk about genotype, those are the genes or the dominant and recessive traits that we put together. Homozygous genotype is the gene combination involving two dominant or two recessive genes. The example is big R or little r. You need to remember that homozygous means same. And when you look at the letters, there are two capitals and two lowercase. Homozygous also means pure. You've heard of a purebred dog. A purebred dog, for example, if you had a golden retriever, that would mean that a purebred golden retriever is where two golden retrievers mate. Heterozygous genotype is a gene combination with one dominant and one recessive allele. This is signified, when we're talking about letters, with one capital R and one lowercase r. So hetero means different. Another term when it comes to hetero is hybrid. And in a hybrid, that means two different groups came together. So if I did my Labrador and a poodle when it comes to dog breeds and mated those two, that's where I get the Labradoodle. Two separate coming together to make one. That's a hybrid. More terms. So once again, this is key to remember. Genotype is the gene combination for a trait. And it comes in the forms of these letters when we're doing our Punnett squares. Phenotype are the physical features resulting from the genotype. So for example, red or white. So these are what you're going to actually see expressed in the particular organism. We can't see the genotype, that's the gene combination. We see the phenotype, which are the physical features. Generation gap. When we're going through our Punnett squares, we always start with the P1 or the parents. So the parent generation is the first group that we're going to combine in our particular experiment. The F1, it's called filial, that would be the offspring from the parent generation. So F1 generation is the first generation offspring in a breeding experiment. This is where we combine two parents, and the F1 generation is our first set of kids, so to speak. The F2 generation, the second generation offspring in a breeding experiment, is called the second filial generation, from breeding individuals from the F1 generation. And you have to remember, when we're talking about doing these Punnett squares, we're not always talking about humans. We can be talking about pea plants. We can be talking about corn. We can be talking about cats, dogs, any type of organism that combines. So we're not necessarily talking about humans. So how does this work? Well, we're going to start with the parents. They are our initial cross. Then, when we cross the parents, we end up with these offspring. So this is our first, F1, our first offspring. We take and we cross two of these, and we get our F2 generation, which is our second set of offspring. So we start with the parents, we move to the first offspring, and then the second offspring. If we're crossing two pure plants, and if you remember, pure means the same letters, the results are all hybrid plants. And when we learn about the Punnett squares in our next podcast, you'll see how these results happen. When we cross two hybrids, meaning we're taking big T times, or big T little t times big T little t, that's where we're crossing our two hybrids, we end up with 3 tall and 1 short plant. And in the next podcast, when we talk about Punnett squares, you'll actually visually see how this is going to work out.